Next up, we have the NCODA Clinical Corner with Annette Paul from Jefferson Specialty Pharmacy in Pennsylvania. Annette?
Hi, thank you. So I just wanted to go over basically a new medication that's going to be on the market. It's called Yonsa. It's a new micronized formulation of abiraterone acetate, which is also known as Zytiga. Next slide. Some of the things I just wanted to go over today were, I just want to do a brief review of metastatic castration-resistant prostate cancer, take a look at where Yonsa's place in therapy is, and also discuss a little bit about the clinical trial that brought this product to market, and look at the safety, and then also go over a little bit how to access this medication. I think it's soon going to be available. Next. All right, so for metastatic castration-resistant prostate cancer, it's really defined as arising PSA while on luteinizing hormone-releasing hormone therapy. The average median time to progression on this therapy is about 23.7 months. And some of the prostate cancer more common symptoms that men usually experience are frequent painful or burning urination, just changes in their urinary flow, new onset of erectile dysfunction, or they could experience some discomfort or pain while sitting caused by the enlarged prostate. So I just wanted to discuss also the timeline of the FDA approval process. So back in May of 2017, there was a new drug application submitted for Yonsa, which was the new novel oral formulation. And in July of 2017, the new drug application was accepted for Yonsa by the FDA. And it was now recently approved in May of 2018 for the treatment of metastatic castration-resistant prostate cancer. And of note, it's to be utilized in combination with methylprednisolone. And the mechanism of action, just to go over this a little bit, abiraterone, it inhibits the alpha hydroxylase and C1720 lyase in the CYP17 pathway. And this helps block the androgen biosynthesis, leading to decreased androgen-sensitive tumor growth. Next. So the Yonsa formulation that will be available, it's in 125 milligram micronized fine particle tablets. This is of note that the formulations are not interchangeable between other abiraterone acetate products. And the total daily dosing that's recommended and has been studied is 500 milligrams once daily. And again, it's going to be used with methylprednisolone. And this dose is going to be 4 milligrams twice a day, which is the equivalent to prednisone twice a day, which is commonly used in combination with other abiraterone products. So currently, Yonsa's place in therapy, it falls in line for use with just the metastatic prostate cancer group. So the other products out there for this indication are Zytiga and Xtandi. Some clinical pearls to take note of with Yonsa is that, again, you should avoid concomitant strong CYP3A4 inducers with Yonsa. It's not FDA approved for metastatic castration-sensitive prostate cancer like Zytiga is, so that's one difference with this active drug and the two different formulations. And it does show to have increased bioavailability with the micronized formulation, which also makes it so it can be taken with or without food. That is one big difference from Zytiga formulation. With Zytiga, you need to fast and take Zytiga 1 hour before or 2 hours after a meal. There's no food restrictions. And while they're taking Yonsa, they should also receive a gonadotropin releasing hormone analog, or they should have had a bilateral orchiectomy. So the clinical trial that was studied was the STAR study, and this was a randomized phase 2 therapeutic equivalent study of abiraterone acetate fine particle formulation versus the originator abiraterone acetate in patients with metastatic castration-resistant prostate cancer. In this study, the primary endpoint was to compare the average serum testosterone levels on treatment days on treatment days 9 and 10 between the two different groups. And again, the treatment groups, they were the fine particle formulation plus methylprednisolone and versus the originator abiraterone acetate plus prednisone. So in this trial, the patients were randomized in a 1-to-1 fashion, and it included men with progressive metastatic castration-resistant prostate cancer, and they were receiving gonadotropin-releasing hormone agonist or antagonist therapy, and they had to have a serum testosterone level of less than 50. The duration of this study were 84 days, and about 88% of the patients actually completed the day 84 visit. Then they pretty much took a look at the safety in this trial in the comparison to the fine particle and the originator abiraterone formulations. They were pretty similar. For the adverse reactions with Yonsa, the most common ones are fatigue, joint swelling or discomfort, edema, hot flashes, hypertension, all very similar to Zytiga. Same thing with the lab abnormalities, can cause elevated liver enzymes. And again, that was just very similar between both groups. So the main highlights from this trial basically show that abiraterone acetate fine particle formulation is therapeutically equivalent to the originator abiraterone acetate. And the therapeutic equivalence was based on testosterone suppression. That was defined as an absolute testosterone level as less than 1. The fine particle formulation was similar, produced similar PSA 50 response rates as the originator. And that was defined as a decrease of 50% in the PSA level from baseline. The fine particle offered a comparable safety profile, and the abiraterone trough levels were similar amongst both groups as well. And then they set up an extension study. This is the STAR-E study. It's an open label, single arm, multi-center study. This included only 20 patients. So again, it's a very small sample size with patients that we're looking at. But they were just trying to evaluate 1-year treatment with Yonsa patients who completed 3 months of either groups of the fine particle or the originator in the STAR study. And then after these 3 months, they basically transitioned these patients just onto Yonsa. All patients in the STAR-E study completed the 1 year on Yonsa. Again, they were just looking at secondary objectives to look at disease progression, evaluation of serum testosterone and PSA levels, and response rates at the 6 and 1 year mark. And again, the safety profile, it showed to be pretty safe regardless what group they started out in, either in the fine particle or the originator group. They transitioned pretty seamlessly onto Yonsa without any issues. So I just wanted to go over a little bit on how to access Yonsa. I did reach out to our distributor, Amerisource, and we were not able to order the medication just yet. I'm sure it's going to become available very shortly. But all GPOs have access to the drug along with any specialty distributor. McKesson is the hub service provider. Patient assistance and copay card programs will become available soon. And Yonsa is approximately 10% lower on wholesale acquisition cost, and up to 25% off invoice discount is available through the GPO contracts. So I was able to locate the NDC, which I provided on the screen for Yonsa. And if you have any additional questions on how to order this medication, I provide the customer service phone number along with the website, which is developing as well. Thank you.
Thank you so much, Annette. And we're going to open the floor again to some more questions, if there are any out there.
Hi, Josh, this is Kendall.
Hi, Kendall.
Hi. Hi, Annette. This is Kendall. I'm one of the project managers with NCODA. I had a quick question.
Yes.
Are there any possible benefits of taking Yonsa over Zytiga that you noticed when you were doing your research?
Well, it was such a small sample size in these trials. So the original STAR study, there was only 53 patients in it. To me, they weren't really highlighting any benefits aside from the being able to take it with or without food. So I see that as being a benefit. I'm not, it's going to be as equally as effective as Zytiga, but I would say for any patients that have issues with, trying to time it around food, this might be a better option.
Thank you.
Thank you, Annette. And then we have one more question for you. How many people were enrolled in both trials?
Oh, yes. So for the first trial, the STAR study, there was only 53 patients, and there was only 20 patients in the STAR-E study. So again, it was a very small population of patients that they took a look at this. And the endpoints, they were evaluated by laboratory assays, basically, between the both groups.
Thank you so much, Annette.
Thank you.